So we have today the first talk by Professor Ramray Bhat of IISc Bangalore. So Ramray is actually a trained medical doctor. So he's one of those rare doctors who actually moved over to research. So he trained in Calcutta Medical College and then went to New York Medical School for his PhD, where he happened to do his doctoral degree under the guidance of Stuart Newman, who happens to be the postdoc of Stuart Kaufman and Brian Goodwin, two names which we will hear a lot about in the school, who are like pioneer modelers in pattern formation. So he comes from a, like really very blue-blooded tradition of mathematical modelers in development. And of course, we have a personal connection because actually Ramray and I were both in the same apartment complex. So it was literally stone's throw, his apartment from ours. So it's a pleasure and he has had actually on multiple occasions appeared in online talks, but this is, I think, the first time he's here in IMSC. So it's a pleasure to welcome Ramray to give his talk on cancer.
Yes, the pleasure and privilege is all mine. Shitabru, thank you so much for inviting me to be part of this workshop. Yes, the lineage is so blue-blooded that I promptly quit development when I could and moved towards a problem that I thought was simpler, cancer. Turns out to be not as simple as I thought it would be. But anyway, so and probably that's the talk that I'll give today. The pain of trying to understand what is cancer. The way I've sort of split this is the 1 hour talk today would be more of a pedagogical exposition of how we can understand cancer in ways that might be distinct from how cancer biologists look at it. So to join the crowd from a more multidisciplinary perspective. And then I will spend tomorrow's 1.5 hour time to talk about some of the work that we are doing in cancer biology, or some of the work my group is conducting over the last 8 years that I have been in the Indian Institute of Science. So I would prefer to do this a little bit more interactive. And so I would, hope for some more participation. Also another thing, it's fun to be following Mohit, especially because he's talked about certain concepts that I wanted to bring back into a discussion, if you will, and then sort of build on some of those concepts and also talk a little bit more about the experimentation, because I'm primarily an experimental biologist. I've been trained in performing experiments to understand the science that we do. So this will be a biology heavy talk, so statutory warnings. Okay, what is cancer? It's not a rhetorical question, it's a question to you guys. What is cancer? Anybody, any thoughts? What comes to your mind? We'll build it from there on. What comes to your mind when we think about cancer? Yes, it's a deadly disease, but no, let's go beyond that. Any special characteristics that makes cancer different from influenza, from hypertension, from stroke? Yes, please.
Like uncontrolled growth of body mass.
Okay, great. 2 phrases, and we can break them down. One is diseases tend to decrease body mass, and cancer or decrease. You said, right? And cancer tends to increase. All right, awesome. If we were to graph the body mass of a patient who suffers from cancer, you would see that there might be a tiny increase, but it's followed by a decrease. So to that extent, it does, and then there are diseases, of course, where you can also have obesity, for example, an increase in body mass and so on. So, but then you touch upon a very interesting concept, and in that second phrase that you talked about, which she referred to as uncontrolled proliferation, I think that was what she meant when she said body mass. In other words, there's this one textbook definition of cancer, which is uncontrolled proliferation of cells. That's there, and that's how it's typically defined in undergraduate textbooks and so on. Okay, so if I were are there any experimental biology students in the audience, or has anybody done and has worked with cells as part of their undergraduate curriculum? Anyone? Yeah, a few of you have. Oh, Ronnie, I can see that. You surely have worked with cells, haven't you? No. Fine. Anyway, some of you have. Okay. The first thing that one would do is when one works with cells is put it in a plastic flask and expect it to grow. Okay. So if I were to take a flask, and this part should be visible to all. Okay. If I were to take a flask where I take normal breast cells, we'll take the context of breast cancer for this lecture, normal breast cells and breast cancer cells, okay, and I were to put these cells in there, and I would give both of these flasks, I would add nutrients to these two flasks. What would happen to these cells as a function of time? See a growth followed by stasis in the numbers over time, right? What would I expect to see in the case of the normal breast cell flask? Ronnie? That's kind of similar to what happens here as well, because after some time, cancer cells also tend to die out because of competition of nutrients and so on. So if I were to compare that with this out here on this graph,
Yeah.
what would you?
So the plateau will be at a lower value of x.
Something like this, right? Okay.
Maybe even lower because it
Maybe even
only has the bottom surface too.
Sure. Okay. Say we stop at a stage where the cells have reached 100% density. Would it look like out here at this stage? This is the 100% confluent stage or density stage. Or would these numbers be different, or do you think it will be like this and then come down like this? Is that what you had in mind? You've put cells in a square unit area. After some time, the cells have covered that area. They've completely covered that area. That's your 100% confluence. Confluence is a word that experimental biologists use to basically define occupancy. You have 100% occupancy now. Okay. So what would you think is happening for normal cells? Okay, let me just take the concept along a little bit. So the first thing that we are seeing is, at least we are arguing in terms of quantity. We are arguing in terms of extent. Do the normal cells divide and reach a state which is out here or somewhere below it? Now, within our body, and these are breast cells that were, say, taken from an adult individual. Within an adult individual's body, do breast cells divide? Don't. If they were to divide, our organs would grow ever larger. If these were liver cells, my liver would be growing larger. Now, why is it that these cells then are dividing? And yes, they do divide. They do divide at a particular rate. Why are they dividing? When they're not supposed to be dividing inside the body, why do they divide out here? True enough. Here I have not given any injury. Even if there is space for them to grow, they will still not grow. There is a lot of space. In fact, our body is full of negative spaces, as one would call it, which is like the space which is unoccupied. There's a ton of space. It's an interesting concept, and we can come to that, but that doesn't seem to be the reasoning here. Yes?
The regulators of the cell cycle is mutated. That's because it's growing.
There is no mutation going on. I'm talking about normal cells here. We presume that there's no mutation going on. Yes, to an extent, yes. To the extent that I'm simply making a point that quantitatively the graph, it is coming somewhere here. Well, I can even challenge by saying there are normal cells which will completely follow the cancer cell and perhaps even reach here. So they are dividing as prolifically, as uncontrollably as cancer cells. Okay? Sorry?
epithelial cells stop the growth of the other cells that is not present.
Great. You mentioned one term, that's a wrong answer, but I will keep this term for later. Epithelial, because Mohit also mentioned this term many times, EMT, epithelial, etc. No, these cells are epithelial cells. Liver cells are epithelial cells. Breast cells are epithelial cells. Epithelial cells also divide. Okay. No, after some time they are together and they are dividing. Some of them are separated and so on. Okay, you mentioned one more term. I will unpack that term subsequently. Contact inhibition. There is a clue here. Yes. Any other answers? And then I'll go on. Maybe because of the way they use their nutrition. There is a certain hint here as well. And the hint lies in the word the way. Okay, let me now perhaps, elaborate my, venture my answers to this. See, the point is we all, as biologists, we work with normal cells and we work with cancer cells. And that allows us, in all the papers that you would, if one were to look at papers in cancer biology, one would therefore always try to compare the normal cells with the cancer cells. Because unless we do that, how do we know what is abnormal about cancer cells? They have nuclei, they have mitochondria, they have Golgi bodies, they have a membrane, they move, and so do normal cells. They divide, and so do normal cells. So by just looking at division in a flask, or by even looking at movement in a flask, this movement aspect was also mentioned some time ago. They do also move in a flask. It is well nigh impossible to differentiate between a normal and a cancer cell. And that's a standard question that is asked in these comprehensive examinations and so on. If I were to take a couple of flasks of normal and cancer cells, how would I distinguish between them? Proliferation is not the key. A fact that they divide, normal cells divide, he tells us that normal cells, that's the reason we can passage them and continue to do so after they were taken, from a patient 53 years ago or 75 years ago. The very fact that we are still able to work with them shows that they must have divided over time. So in other words, cell proliferation seems to be some kind of a universal property of cells. It almost feels as if the default state of cells is to divide. Yes, Shitapu.
Yeah, but you can, just by looking at cell morphology, distinguish between cancer and normal cells, right?
A great point, cell morphology. I want to look at, sorry. Yes, I'm going to continue with that. Okay, if I were to look at this, now I was looking macroscopically. If I were to look at this under a microscope, breast cells are looking like this. Normal breast cells are looking like this. Cancer cells are looking like this. I mean, humor me when I say this, that it's not easy to distinguish based on cell morphology, again, by looking at these cells, which one is what. By nucleus, we can distinguish it. No, we cannot. If by cell shape only we can't distinguish it, by nucleus also we can't distinguish. There are some aspects, so I give you, there are some very specific aspects by which we can distinguish it. Yes, Rahul, sorry. You can get back. I'll talk about that.
So can we distinguish them by stiffness? So cancer cells seems to be a bit stiffer than the normal.
Do you mean the stiffness of the cells itself?
Not the cells, but the cluster of cells.
By the cluster of cells. Okay. Okay. Stiffness. We can get to stiffness as well if there is time now. Okay. Or else I can talk about stiffness tomorrow as well. All right. I'll move ahead.
Also, I was just going to ask about, you mentioned we are working with cell lines from 50 years ago and 75 years ago. These are all either they're cancer cell lines or they're immortalized, so normal cells die, right? So the apoptosis.
These are immortalized. So yes, right. To that extent, they have sort of gone past that Hayflick limit.
I mean, I don't know what the answer here is, but my impression was normal breast cells will eventually die off. To keep them alive, you have to
immortalize them. So there is the introduction of a particular viral gene that will allow them to therefore evade senescence after a while. I buy that point. And that comes to the telomerase aspect that, to that extent, but that's the point again. These normal cells are also immortalized. And so on that respect also, they are indistinguishable from cancer cells. But where lies the difference? If I take these cells and put them within a three-dimensional environment, an environment where they don't see plastic, and there lies the point of the stiffness as well, which we'll come to in some time, where they see themselves in the same environment in which they would see themselves in the breast or in the liver of the individual, there things start becoming different. I've taken the same cells and now I put them within an environment, within a proteinaceous environment. Instead of putting them on plastic, I put them in a proteinaceous environment where, like I'm drawing a cuboid of the kind of proteins that you would tend to see inside the organ, and here the cancer cells would start forming structures like these, whereas the normal cells would start forming structures like these. This represents a gland. It can be a mammary gland or a breast gland, where if you give the right kind of hormones, it will start secreting milk proteins in here. So it, in other words, replicates the structure and function of its physiological self when placed in the right biochemical and biophysical microenvironment. On the other hand, in that same breast microenvironment, you put the breast cancer cells and they start doing what they would otherwise do also, moving, breaking down, degrading, dividing. So the uncontrolled proliferation of cells definition has to have that additional phrase of being in its right native microenvironment.
It appears that since you said that when the cancer cells go into this tissue, they start doing the same thing as they were doing before. So
Yes.
So the addition thing is to look for in the normal cells, which seems to be lacking in the others.
Yes. Or one can also flip it by saying that the normal cells in its non-native environment does behave very similar to a cancer cell, which comes to the question of, I think this was also brought out in Mohit's talk, what is normal?
Right.
What even is normal? Now, again, what differentiates a normal cell from a cancer cell? A normal cell can very well behave very similar to the cancer cell. Now, I'm talking about now, of course, maybe 19th century biology, looking under a compound microscope and looking at the cells. Let's break this down into molecules. So, if one were to look at signaling, I don't have flags and landscapes in my talk, because signaling is there. If one were to look at signaling, PI3 kinase, MAP kinase, two signaling features which are commonly upregulated in cancer cells. Okay, PI3 kinase, MAP kinase, AKT. There are a bunch of molecules. I can name them, but that's not the point. The point is these are signaling pathways that typically end into the nucleus and turn on cell cycle regulators that would allow the cells to keep replicating. If one were to look at normal breast cells in that flask and try to look at these signaling pathways, one would see them being upregulated. And so it's not just at the level of cell behavior, at a gross cell behavioral level. One can even see that in signaling studies. One can see that in molecular studies where you can see MAP kinase, a phosphorylated MAP kinase molecule moving into the nucleus, which you would also see in cancer cells, which you don't see in a normal cell that is part of, say, a biopsied specimen of a normal breast cell. But you take this cancer cell, take the normal cell and put it in its environment, now you don't see the MAP kinase being activated. Now you don't see the PI3 kinase being activated. You don't see the AKT being activated.
The organ cell, the kinase level that you will see. So in a cancer cell, it will be like higher. It will be a
No,
higher.
it would be the same in the flask. In the flask, the MAP kinase activation in a normal cell is equivalent to the MAP kinase level activation in the cancer cell.
So what is being different in the signaling?
In the flask, they both are in that same plastic environment, and hence they behaviorally they are similar to each other, and even at the level of molecular signaling, they are similar to each other. But now you take them and put them within their native environments in a three-dimensional microenvironment. Now your MAP kinase levels are high here, PI3 kinase levels are high here, proliferation is high, invasion is high, production of matrix metalloproteinases, which are enzymes that cut through the environment, allowing for cancer cells to move through, is high. In these cells now, MAP kinase levels are down, PI3 kinase levels are down, level of MMP activity is down, and these cells are no longer dividing, these cells are no longer moving, and hence we get this quiescent homeostatic architecture.
So is this something to do with constitutive activation? Like
No, it's not constitutive activation, therefore it is contextual activation of signaling driven by the microenvironment. So in other words, what I'm telling you is that cancer represents a state of cells, as of now, of cells where the context determines whether the cell will behave in a cancerous manner in terms of uncontrolled proliferation and migration, or whether the same cell can behave in a quiescent, sessile manner where it does not divide or does not migrate. Is the cues for its behavior is coming from its surrounding.
Okay. I just wanted to understand.
Yes.
So you're saying that the environment matters to
Yes.
more than any intrinsic whatever expression levels and so on.
So I'm building a sort of a genotype environment map of how a cell behaves as of now.
Okay. But the environment itself is shaped by the presence of these cells, right?
Fair.
So
Fair.
it's a feedback.
Yes, there is a feedback. So there is a dynamical and reciprocal feedback that comes in. I will talk about the feedback. Yes.
So taking a part of breast cells and
Yes.
then putting them somewhere in the liver,
Yes.
will they proliferate into cancer?
Brilliant question. That's been done by Gil Smith at NIH, who did a lot of these lovely experiments. And what he found out was that in many cases, if there's some trickeries, of course, that experimental biologists apply, but with some tweaks and so on, the mammary cell can be differentiated now into a hepatocyte or a liver cell in the liver microenvironment.
Yes.
Okay. So he took stem cells, and so because he gave it some potential for it to divide. So if you give it some potential for it to sort of change its differentiation fate, allowed some level of plasticity, because a cell that is differentiated is very difficult to sort of pull it out and bring it into another environment. But if you put it in another environment, it will take the fate of that environment, and not just that, it will get integrated with the liver environment and behave like a liver cell. So that's been done. Yes.
So,
Yes.
so as I understand, pretty much a similar question to her.
Yes.
So if you take a cell which you think is cancerous in somebody who is a cancer patient, you take out that cell and try to embed it in a person who's fine.
Yes.
If one were to imagine such a thing,
Yes.
that person wouldn't catch cancer, it seems.
Right. So a kind of this experiment was done in 1970s by David Dahlberg and Mina Bissell. She was my postdoctoral advisor. It was not the exact experiment that you suggest, because that's a little harder to do.
Yes.
But what was done was that this was done in chickens. So in chicken embryos, because at that time chicken embryos were could be studied very easily, because you can manipulate a chicken embryo and actually watch it within the egg. What they did was they overexpressed a particular oncogene. Oncogenes are genes that when get mutated, turn on the cell cycle regulators towards allowing cells to divide fast. Okay, so the most famous of these genes are called RAS oncogene. The RAS oncogene is a gene. That basically, the point being that if you have a mutation in that gene, cells start dividing like a cancerous cell. They put that RAS oncogene into the wing cells of the chicken and watched it. In an embryonic state, nothing happened. When the chicken became an adult hen or whatever, it started dividing. Other words telling that an embryonic microenvironment discourages, even if you have the right kind of oncogenes or right kind of genotypic state for a cell to divide, if you have an inhospitable microenvironment, it will prevent the cancer from spreading.
But later it did spread.
But yes, once the environment has changed, because an embryonic environment is very different from an adult environment. Kind of, harks towards why we get cancer more as we grow old also. But that's for a different lecture. I'll talk about that tomorrow. Yes. We can.
We can say, and then epigenetics matter.
Well, I can also say cells matter. I mean, it's just, these are terms, right? You are absolutely right. To that extent, there is, I'll sort of, pay respect to the point of epigenetics matters by saying that you don't need mutations to get cancer. Gene expression can also get you cancer if the microenvironmental insults get transmitted instead of a mutation taking place. To that extent, yes, epigenetics matters. Yes.
That you started off with.
Yes.
But now having the microenvironment, at least partially, some elements of it, what is the difference that we would see?
Right. So the difference that you would see, so in the flask, is it? Are you asking about the flask? So in the flask, what you don't have is this proteinaceous matrix that surrounds the cells in those cuboids, and that's what instructs the cell. So thank you for giving me the cue. Let me now build the idea from a cell onto a tissue state. Okay. So just maybe hold on for like 5 minutes, and then we'll again take the questions. Okay. So you would see that I've drawn a multicellular state here. Okay. Not a single cell state. Most of the things that I was talking about can well relate to a cell, right? So I've drawn a multicellular state here and a multicellular state here. Where is multicellularity coming into this picture? I was talking about microenvironment, MAP kinase signaling, cell proliferation, cell migration. Why is it that the cells, they could all sort of stay in this cuboid as single cells and do what they had to do? Why did I draw this here like this? And there's another angle with which we can enter here. If you remember Mohit's slide on EMT, again, Mohit draws his epithelial cells like this. And then he drew his mesenchymal cell as something like this. The reason why his cartoon had these shapes, an epithelial cell was not something like this, and mesenchymal cell was like this. There's a reason. What's that reason? What's the reason I too have drawn my cells here as, cubes to columns kind of structure, whereas out here, oh, that's gone now, the cancer cells were like all over the place. Again, microenvironment. Microenvironment, what it does now at the tissue level. So let's start building the molecules and the cell biology together. Oh, darn, I've rubbed all the T terms. That's okay, we'll come back to those. Let's start building what the microenvironment is doing. So the question to, what is it, what was not there in the flask, what's there in that cube, three-dimensional cube, that allows them to behave how they have to behave. What you have is, so now we are looking at this in a sort of, a wide arc, a small arc of a wide circle, big circle. So if you look at these normal cells as they exist in our body or in that cube structure, what is it that's going on out here in molecular terms? What you have is this matrix which is present out here. Now this matrix consists of proteins. These proteins are extracellular proteins, and therefore they form matrix-like structures, network-like structures, and therefore they are called extracellular matrix. This matrix basically connects with these cells via receptors such as integrins. Names are not very important here for the purpose, but since I'm discussing them specifically, I'll just put in the specific terms. Via integrins. Now, what happens subsequently is that when these linkages come together, which don't happen in the flask because there is no matrix in the flask, what now happens is this signaling tends to do a few things inside the cells. A, it tends to, so let's put it this way, integrin ECM, okay, once this linkage is in place, it tends to inhibit MAP kinase phosphorylation. In doing so, it tends to douse proliferation. It also tends to inhibit a different set of integrins which are involved in migration, proliferation. In the process, it tends to make cells less migratory. And there is also crosstalk here between the integrins and MAP kinase, which is positive in nature. So if these two are present, they would push each other, and that's why proliferative cells also tend to be migratory, and vice versa. Now, what it does is it tends to now induce the expression of E-cadherins. What are these molecules called E-cadherins? These are molecules which sit out here between the cells and tend to get the cells to adhere with each other. And in this process, what you see is the microenvironment is getting the cells to tie up with each other, not proliferate, not migrate. And that, along with a few other things that it also tends to do, it tends to send, once these links are in place, this signaling tends to send certain proteins to this edge and certain other proteins to this edge. In this process, it sort of builds up this architecture that allows intercellular adhesion, cell matrix adhesion, a structure that comes through, and this structure is what we call an epithelial cell. This is an epithelial cell, and therefore an epithelialness is not a cellular property. And this comes through, again, developmental biology as opposed to cancer biology, where cancer biology, given its obsession with unicellularity up until about maybe a decade back, when collective invasion became very fashionable, where cancer biology tends to think about EMT, or epithelial to mesenchymal transition, or epithelial to mesenchymal whatever, as cellular properties. They are not. They are tissue properties. Because an epithelialness not just involves being a cell that has to remain static and non-migratory, it also has to tie up with each other and form these multicellular structures. In fact, it's not just this. If one were to try and tease apart, and these are biophysics experiments, when one wants to sort of tease apart a structure like this in order to find the magnitude of what is the stiffness of this structure. So stiffness was mentioned. How stiff is this structure? One would see that the stiffness of this entire multicellular structure cannot just be understood through the strength of the bonds between the E-cadherin. So now what am I talking about? What I'm saying is there's much more structure to this kind of a epithelial nest. What gives strength to such structures are also actin cytoskeleton, which are like cellular cytoskeletons, which typically tend to be inside cells. So again, we tend to think of them as inside cells. They give rise to these structures. But what is happening here is when this kind of structure forms, you have these actins which start linking across the cells and form these additional cables which make the structure very strong. Now, what I'm talking about here is relevant to embryogenesis. It's also relevant to homeostasis. And it is these actin structures which move across, which are sort of mesoscopic in their scale. They move across the cells, which gives rise to tissue level property. These actin level, these supracellular actin cables also contribute to epithelialness. So epithelialness or epithelial cell cannot just be defined on the basis of certain markers which keep going up and certain markers which keep going down. One has to look at the tissue level properties. All right. So I think there were a few hands raised. Yes, let's take some questions. Yes. Yes, this is at the level of organs. If you call organs as a bunch of cells, then yes, it is at the level of organs. To the extent that if you were to stain for actin in that structure that I showed here, right, you would, and but these are the individual cells, right? So I would, in a classical context, expect to see the same level of actin out here, out here, out here, and out here. What I see though is there's an immensely strong, continuous layer of actin which is out here. And this actin cable, which perpetuates and along with the coherence sort of builds up, gives strength to the structure. Yes, absolutely. Well, okay. If you were to talk about the breast as an organ, the breast as an organ would consist of thousands of such ducts. So you would see it in all the ducts. You would obviously not see it across the organ because an organ doesn't consist only of cells. It has the matrix between it as well. So between the units of the organs, yes. But in the tissues, yes. Okay. Yes, Ronnie.
Can one decouple the contribution which comes from integrins and cadherins?
Great question. Since the word contact inhibition was mentioned, this state is precisely what is contact inhibition.
Yes.
This is the molecular mechanism for contact inhibition. When you have e-cadherins linked to each other, cells are not dividing. But now we know why. And now I can trace the molecular pathways. Up until now, we couldn't understand that why is it that when cells come close to each other. So the word is contact inhibition. Contact inhibition is a term that is used to explain why when cells come next to each other, normal cells come next to each other, they zip up. It's because of this. Because there is also another feedback from e-cadherin onto this linkage. Okay, so e-cadherin also gives rise to this structure. And because of this, when cells come close and their e-cadherins link up, you again get a reinforcement of this entire process, leading now to quiescence and non-migration. And that's why normal cells get zipped up. That question then, let me ask, what makes now normal cells different from cancer cells? In cancer cells, what you have is you have e-cadherin which is now mutated such that it's no longer sitting on the cell surface. So there is no feedback. You have MAP kinase which is driven through this particular protein called RAS that is perpetually activated. So there is no quiescence going on because even if you can have the integrins linked to ECM, there is no inhibitory loop that stops this from going to the nucleus. You have delocalized integrins because of other mutations that prevents this loop from going down to the integrins. And hence, in a cancer cell, even with the right context, and hence that cube of the cancer that I showed, where even in that native environment, the cancer cells tend to migrate and proliferate, is because these feedbacks are all wiped out due to mutations. Or, if you don't give it the right context, of course you will again have the state come through, and the normal cell also starts behaving in a proliferative, migrative capacity. MAP kinase also gives rise to MMPs. Yet another feedback. I think Mithun was the person who asked about this. MAP kinase also tends to give rise to MMPs. MMPs, remember, are those protein cutters which destroy the microenvironment. They basically degrade ECM. Okay? No. So again, in a normal cell, integrin ECM connections will lead to decrease in MAP kinase phosphorylation, will lead to a decrease in MMP activity. In a normal cell, which is in a flask, no ECM, increased MAP kinase, increase MMPs. In a cancer cell, in whatever environment, no effect of this on the MAP kinase pathway, increase in MMP. MMP in turn starts degrading ECM, leading to a more vicious cycle, leading to immense exploration of the surrounding by the cancer cells. And hence, again, the experiment would be, if I take a normal cell and upregulate MMP, it will start behaving like a cancer cell. Yes, Ron.
Talking about the chicken embryonic experiment earlier. So in that case, was it that in the embryonic stages, integrin alpha was sort of not that highly expressed? So even if you have RAS overexpression, the activity was...
Yes. Any other questions? Yes. Sorry? Excellent question. How do you differentiate between a cancer cell? What is a pathologist really looking for? So a pathologist, when she looks under a microscope, she's basically solving a problem of pattern formation. So pathologist also is a pattern formationist or whatever. So when you look under a biopsy, you're basically looking for this square and the misshapen circle. That's all. So if you see that the cells are square shaped, you know it's not a cancerous specimen. And if the cells are rounded, it is cancer. That's all pretty much. It's just a very deep learned understanding of cell shapes in a population that a very smart intuitive pathologist sort of builds her intuition on. I'll just add one more feature since you asked this fantastic question. If you were to see a pathologist's report from a hospital or from a laboratory, you'd often see this term called grade. It's a grade 2 tumor, it's a grade 3 tumor. Again, a grade is nothing but a pattern formation metric. A grade basically tells you the level of de-differentiation from what should be the typical shape of a cell to some kind of a shapelessness. The higher the grade, the more dangerous the cancer, the greater the departure from the native architecture.
So the ability of the normal cell to kind of grow to an organ is mainly because of the dimensionality of the whether it's 2D or 3D, because even in 2D culture you can see the normal cells adhering because of the N-cadrins and E-cadrins. Even if you see the expression levels, you can see the N-cadrins and N-cadrins present. Isn't that because of the dimensionality why it's forming an organ rather than...
Is it the dimensionality, or is it... So I tricked in the aspect that when I talked about dimensionality, I did not also talk about the sort of the biochemistry, and I'll elaborate this a little bit. So is there a way, therefore, that I can get cells in a two-dimensional environment to also behave in a non-migrative, non-proliferative capacity? Okay? If that's the question you're asking, because the flask was two-dimensional, right? So I can. And the way I can is by having the right kind of matrix on the surface and also giving it the right stiffness, the right elastic and viscous modulus. So now we come to the point that one cannot now put these normal cells in any environment, any three-dimensional environment, and expect it to behave like a normal cell. You need to have the right biophysical properties and the right biochemical properties for those cells to now become quiet and not migrate. Okay?
All these signals that you mentioned over there, I want to know which of these are actually dependent on just the environment and which have to do something with the cell. So it seems as if this is entirely, right now, it seems as if it's entirely the environment's game.
Entirely the?
Environment's...
No, it's not. Remember, that's why I drew this pattern as a means of showing you how what she said about epigenetics or what a cancer biologist says in terms of mutations can all be accommodated into this idea. Let me belabor this point. You have the environment. The environment keeps the signaling in check. The signaling keeps a modulator of the environment in check. And because of this, the entire state remains in a quiet state.
Okay.
Now, what can happen is, if you have the wrong kind of environment that sets the signaling towards a cancer-like behavior, that leads to a modulator of the microenvironment getting turned on, which goes and breaks the same environment, leading to a cancer-like state. Okay, that's your epigenetic idea. A genetic idea would be, whatever your environment might be, a chance mutation in the signaling pathway leads to the signaling pathway becoming independent of the environment, leading to an upregulation of the signaling towards a cancer state, leading to a modulator of the environment now coming through, which breaks the environment.
Okay. So mutations is the...
So your environment, your epigenetics and your genetics all are linked under the same signaling network. But the point I'm trying to again build is, the signaling network is not that of a cell, it's that of a tissue. It's that of a tissue. Because again, I can break this not using a cellular feature like say the MAP kinase, but using E-cadherin. If I take the E-cadherin and alter it, that can again lead to changes in the cell ECM.
I'm sorry, I'm not a biologist.
Never mind. What is E-cadherin? E-cadherin is that which links cells to each
that's
other.
Okay. But what is
In
the
other
source
words,
of it?
a modulator of a tissue state can also drive the state towards a cancerous state or a normal state.
Yes, I understand. It's providing the force for them to be together. But the question is, what is the source of it? Again, it's the environment, right?
No, the source of it, the source of this protein is the cell.
Is the cell. Okay,
the cell.
I see.
The cells are making all the proteins that are required. Even this environmental protein is being made by cells. So the cells in the microenvironment are linked in a harmonious fashion as long as you don't have alterations in this environment from an extraneous environment, such as say wound healing. Why is it that often tumors are associated with wounds? Because what has happened in a wound is that this linkage is now coming under threat. Okay, so I think there's this biologist called Dvorak who said that a tumor is a wound that never heals. Fanciful statement, but whatever. There's some logic there that's hidden, and that is here. The extracellular matrix, then this particular linkage is what can get altered in a wound. And if you have the wrong kind of genetics or some kind of insusceptibility here, you can lead to this linkage now getting broken, leading to this state again coming through. So therefore, the wound or wound healing aspect also comes into the picture. Okay.
Is it like the mutation?
Okay. Let's talk about the environment. If I were to think of this as, say, for example, a lung cell, okay, how would I connect smoking with this state? Now let's talk about an completely macro environment. I was not talking about the macro environment. I was just talking about the environment, a biological environment around a cell. What happens when, say, you have pollution or smoke coming through? So the environmental cause of cancer. Can we link that also to these molecular mechanisms? Yes, we can. How? So, say if you have smoking or pollution coming through, how can we link these to the molecular mechanism? Smoking or pollution typically brings from outside that organ, maybe we should start drawing from this side, from outside the organ, a bunch of other cells such as the immune cells. So Mohit mentioned these macrophages in his talk, right? The the macrophages which are involved in gobbling cells and so on. So these are immune cells. Immune cells have one important function in the body, and that is to basically restore this ECM. So when a wound heals, it heals because immune cells come and build this matrix around the cells, hoping that, look, I have built my ECM. I hope this is restored. I hope the organ behaves in the way it has to behave. Okay? When you have too much of pollution or too much of smoking, what would you get is too many immune cells coming in. And too much is as bad as too little. So now you have excess ECM. Excess ECM is also detrimental towards a harmonious connection between the cell receptors and the ECM, leading to, once again, a disruption of this cycle, leading to the cell going towards a cancerous state. And now, therefore, we can build into our theory of how the macro environment disrupts the microenvironment, disrupts the signaling, disrupts the epigenetics, disrupts the genotype or the genome or the gene expression or whatever, epigenome, leading to a cancerous state.
Cause the cancer because it will actually repress the pathway. And
yes, to a great extent, yes. Well, I'm simplifying things a little bit here. What you have is this ECM itself is a complex of a bunch of different molecules. You need to have the right ratios of those molecules to give you this sort of a superstructural sheet on which the cell sits. Actually sits on a sheet of these proteins. But now when these immune cells come and they in turn react to the pollution and the smoking and so on, what they do is when they produce excess amounts of this extracellular matrix, often the compositions or the ratios of which these proteins are is disrupted. And that leads to this sheet not forming properly. And that leads to those linkages.
One more thing,
yes,
is that why the cells are a bit stiffer? The cluster of cells are a bit stiffer.
Yes, precisely. They secrete a particular protein called collagen, which is involved in strengthening our tendons and so on. And that collagen tends to be stiffer. The matrix that forms this sheet does have collagen, but that's not the only component. But these immune cells come and make so much of collagen that's what makes the tumor stiffer, which is why, again, for breast cancer, it's always important for women to do self-examination, find lumps. That lump is because of the stiffness caused by excess collagen that is being produced by, say, immune cells and so on.
So,
yes.
So when we say that microenvironment is what is guiding the cell fate, basically cancerous state. So is it possible to basically convert a cancerous cell into a
yes, it is.
Yes,
it is. So there have been studies, unfortunately not completely in the treatment domain, but there have been studies which if you take a cancerous cell and if you were to sort of disrupt any of these points where there is an excess, RAS leading to MAP kinase, extracellular matrix leading to RAS, etc., all of these, if you are able to disrupt the signaling, you can actually get the cancerous cell to now take this shape and form these kind of structures. You can.
It's basically if we restore the cell.
Yes. The tissue state. Yes.
Well, sir.
And that's the reason why these therapies fail. So the most sort of exciting therapy based on this particular idea came out in terms of MMP inhibitors. So towards in the early 2000s, in the early 2000s, MMP inhibitors were tested as a great breakthrough for cancer therapy, because the idea was simple. If you inhibit MMP, this whole thing can probably shut down, right? Spectacularly failed, because again, I think as Mohit explained, there's so much common between development and cancer, and you can see that from what I have been talking about, that if you were to inhibit MMP, you will inhibit every cell that uses MMPs to shape our body. Our body is constantly being shaped by cells such as fibroblasts, which secrete matrices and are constantly healing, repairing, moving around, shaping, reshaping our body. And if you were to use MMP inhibitors, all of those processes will stop. And as a result, we will probably die of something much earlier than of cancer. So yes, many of the problem of using ways to tweak this signaling is that this signaling is intrinsic to our homeostatic state. So if one were to use disruptors of such signaling, one runs the risk of having side effects galore within our body, and that has serious consequences.
So what is the popular procedure right now? I mean, that is used to tackle this?
So the popular procedure still in most cancers happens to be brute force chemotherapy, which is that you use agents that forget all of this and go right into the heart of the nucleus, where they intercalate within the DNA and sit there such that if the cell divides, which cancer cells divide, if the cell divides, then it will basically cause so many DNA damage points that the cell will die. And that's the way chemotherapy kills the cancer cells. That's the same reason why chemotherapy also kills every other dividing cell in our body, such as the hair follicle stem cells, which is why we lose hair, such as the stem cells sitting in our intestines, which is why we tend to vomit or have diarrhea when we take chemotherapy, and so on. Okay, so there are so many cells in our body which are constantly dividing. Chemotherapy goes and hits those as well.
So it's not much targeted?
Absolutely not. So that's why there is some targeted therapy that is coming in. For instance, the targeted therapy which is used in breast cancer targets this link. So there is apart from the integrin, there is also a link with a particular receptor called EGF. So EGF receptor. So there is a particular so this turns on this and this turns on the MAP kinase pathway. I didn't want to add too much details. So what they use is an antibody that goes and binds to this receptor and turns this receptor down and therefore breaks this particular cycle. And that's called the HER2 therapy, Herceptin, which is used in certain breast cancers where you have an upregulation of this receptor. You can turn the proliferation down using the signaling state. Okay.
But that is targeting the MAPK signaling.
That will target eventually the MAPK signal.
But it will also affect the normal cells also, right?
Thank you for asking that question. So that's the difference. That's the reason why Mohit drew his E cell like this and his M cell like this. Because you have actin cytoskeleton shaped like this, and that's what leads to the square to rectangular state. If you were to examine actin out here in these kind of cells, the mesenchymal cells move in this manner, and you have excess of actin here and an excess of actin in these sort of extensions of the cytoskeleton, and here you would see actin present. And this actin is involved in basically pushing the membrane further along for the cell to migrate further. So the cytoskeleton is very differently structured in a mesenchymal cell as opposed to an epithelial cell. And that determines the shape of the cells, making a cell epithelial to mesenchymal. That also tells us, interestingly, that one can use actin regulators or cytoskeleton regulators to perhaps move cells from an epithelial state to a mesenchymal state. It also tells that perhaps the body can also do the same thing. And that's why in the body, we have multiple switchings between an epithelial to a mesenchymal state as a cell moves. In fact, in that sense, just to add one point, therefore the default state of a cell is also to move. A cell, given space, would move. Keeping the cell sessile or in a non-migratory state requires work, requires an active signaling axis. Just the same way that the default state of a cell is to divide, and keeping a cell quiescent is an active property of the tissue, and it actively works, an organ actively works to keep its cells in a quiescent, non-proliferative state. Okay, And so therefore, again, anti-proliferativeness or, quiescence and anti-migration or sessility is a tissue property, not a cellular property. Therefore, cancer, forget being a genetic disease, is a tissue disease, not even a cell disease. Okay, that's the sort of take-home message that I want to drive in this talk, and I think I've run out of time. So thank you.